Good day everyone. I'm Kendall Loizaga and I am your reporter for today. For today's lesson, I will discuss to you it, so we have three topics that I will discuss. So first is synthesis and processing of messenger RNA, or what we call the mRNA. Second one is the encoding of genetic information, and the third one is the translating of genetic information. So let's discuss first the synthesis and processing of messenger RNA. Transcription is the DNA-directed synthesis of RNA, and messenger RNA is transcribed from the template strand of a gene. RNA polymerase separates the DNA strand at the appropriate point and binds the RNA nucleotide as they base pair along the DNA template. So like DNA polymerases, RNA polymerases can add nucleotides only to the 3 end of the growing polymer. So genes are read to 3 to 5 or creating a 5 to 3 RNA molecule. Specific sequences of nucleotides along the DNA mark where gene transcription begins and ends. So we call the begins of transcription as promoter, and the end of transcription is we called as the terminator signal. So bacteria have a single type of RNA polymerase that synthesizes all RNA molecules. In contrast, eukaryotes have 3 RNA polymerases 1, 2, and 3, and their nuclei. And one of the important in mRNA synthesis is the RNA polymerase 2. So it is used for mRNA synthesis. Transcription can be separated into 3 stages: initiation, elongation, and termination. First is initiation. During initiation, a section of DNA called promoter DNA section signals for and when transcription takes place. The protein RNA polymerase binds its promoters and separates the double-stranded DNA. Once it's separated, the RNA polymerase binds to the DNA and begins transcribing a section on one strand, the template strand of DNA, into RNA. The promoter identifies section to be copied but is not copied itself. After initiation, so we go to elongation. Elongation occurs as the RNA polymerase read the template strand and build a complementary RNA strand by bringing the appropriate nucleotides together. This RNA strand almost identical to the non-template DNA strand except the thymine and DNA are replaced with uracil in the RNA strand. So when say in termination, there is a section of DNA to signal where transcription starts, and there is a section of DNA signal where transcription stops. The section of DNA is called terminator sequence. Once the RNA polymerase reaches the terminator sequence, and the RNA strands separate from the DNA. After termination, the new reconstructed RNA strand undergoes some moderation to remove sections that will not be translated. The RNA then leaves nucleus to be translated into protein. So those are the 3 processes of transcription. So continue. The presence of a promoter sequence determines which strand of the DNA helix is the template. Within the promoter is the starting point for the transcription of a gene. The promoter also includes a binding site for RNA polymerase several dozen nucleotides upstream of the start point. In prokaryotes, RNA polymerase can recognize and bind directly to the promoter region, while in eukaryotes, proteins called transcription factors recognize the promoter region, especially a TATA box, and bind to the promoter. So as you can see to the figure, after they have bound to the promoter, RNA polymerase binds to the transcription and factors to create a transcription initiation complex. And RNA polymerase then transcription starts. As RNA polymerase moves along the DNA, it untwists the double helix 10 to 20 bases at a time. So take a look on the figure again. So the enzyme adds nucleotides to the three end of the growing strand, and behind the point of RNA synthesis, the double helix reforms and the RNA molecule peels away. A single gene can be transcribed simultaneously by several RNA polymerases at a time. A growing strand of RNA trails off from each polymerase. The length of each new strand reflects how far along the template the enzyme has traveled from the start point. The congregation of many polymerase molecules simultaneously transcribing a single gene increases the amount of mRNA transcribed from it. This helps the cell make the encoded protein in large amounts. Transcription proceeds until after the RNA polymerase transcribe a terminator sequence in the DNA. In prokaryote, RNA polymerase stops transcription right at the end of the terminator. So like what I said before, terminator is the end of transcription. It is both on the RNA and DNA then released. In eukaryotes, the polymerase continues for hundreds of nucleotides past the terminator sequence AAU-AAA. At a point about 10 to 35 nucleotides past this sequence, the pre-mRNA is cut from the enzyme. Number 2. Eukaryotic cells modify RNA after transcription. Enzymes in the eukaryotic nucleus modify pre-mRNA before the genetic message are dispatched to the cytoplasm. At the end of the pre-mRNA molecule, a modified form of guanine is added, the 5-cap. This helps protect mRNA from hydrolytic enzymes, and it also functions as an attached shear signal for ribosomes. At the 3 end, an enzyme adds 50 to 250 adenine nucleotides, or what we call the poly-A tail. In addition to inhibiting hydrolysis and facilitating ribosome attachment, the poly-A tail also seems to facilitate the export of mRNA from the nucleus. The mRNA molecule also includes non-translated leader and trailer segments. So, the most remarkable stage of RNA processing occurs during the removal of a large portion of the RNA molecule during RNA splicing. Most eukaryotic genes and their RNA transcript have a long non-coding stretches of nucleotides. Non-coding segments introns lie between coding regions. The final mRNA transcript includes coding regions, exons that are translated into amino acid sequence plus the leader and trailer sequences. So, as you can see on the figure, RNA splicing removes introns and join exons to create an mRNA molecule with a continuous coding sequence. This splicing is accomplished by spliciosome. When we say spliciosome consists of a variety of protein and several small nuclear ribonucleoproteins, or what we call SNRNPs. Each small nuclear ribonucleoprotein has several proteins molecules and a small nuclear RNA molecule. or snRNA, and it is about 150 nucleotides long. So what you can see to the picture, number 1, pre-mRNA combines with snRNPS and other proteins to form its spleosome. So it is pre-mRNA and small snRNPS are combined. Number 2, within the spleosome snRNA base pair with nucleotides at the end of the intron. And number 3, the RNA transcript is cut to release the intron and the exons are spliced together. The spleosome then comes apart, releasing mRNA, which now contain only exon. Okay? In this process, the snRNA act as ribosome and RNA molecule that function as an enzyme. Like pre-mRNA, other kinds of primary transcript may also be spliced, but by diverse mechanisms that do not involve spleosome. In a few cases, introns RNA can catalyze its own excision without proteins or extra RNA molecules. The discovery of ribozymes rendered obsolete the statement all biological catalysts are proteins. So remember that. All biological catalysts are protein. So in RNA splicing, it appears to have a several functions. First, at least some introns contain sequences that control gene activity in some way. Second one, splicing itself by regulate the passage of mRNA from the nucleus to the cytoplasm. And the third one is, one clear benefit of split gene is to enable a one gene to encode for more than one polypeptide. Alternative RNA splicing give rise to 2 or more different polypeptides depending on which segments are treated as exon. Its results of the Human Genome Project indicate that this phenomenon may be common in humans. Split genes may also facilitate the evolution of new proteins. Proteins often have a modular architecture with discrete structural and functional regions called domain. In many cases, different exon code for different domains of a protein. The presence of introns increases the probability of potentially beneficial crossing over between genes. Introns increase the opportunity for recombination between 2 alleles of a gene. This raises the probability that a crossover will switch one version of an exon for another version found on the homologous chromosome. There may also be occasional mixing and matching of exons between completely different genes. Either way, exon shuffling could lead to new proteins through novel combination of function.